Thank you everybody for coming. For those of you who don't know me, I'm Geraldine Wood. I work at Massey. I'm also a member of the Manawatu Orchid Society, and at Massey is where I met Carlos when he came from Chile to do his master at first and his PhD, and he loves New Zealand so much he's come back. So yippee! And better still, he's doing work on our native orchids, so that's another project that's dear to my heart, given that the New Zealand Indigenous Flora Seed Bank has been set up, and Craig actually wants to get our New Zealand native orchids into that seed bank. So Carlos is the man of the time. He's done a lot of work on Gastrodia, and so he'll teach us the principles of what we need to do in our
Thank you very much, Geraldine. Thanks. Just get my glasses on so I can see who's sleeping. It's actually a great pleasure to be here today and get to talk to you about the research I'm doing at Te Papa at this moment, or now, on orchids. And it's actually, it's a great venue. I've always been a very keen, well, very interested in the greenhouse in here, and also the esplanade. This is sort of like the way that I took every time that I was going to Massey University from Savage Crescent, where I used to live with Viv, and every morning walking up to Massey to do my studies, my master's first, and then my PhD. I've been in New Zealand since 2000, so it's been about 13 and 14 years, and you can tell that my accent, Chilean accent, is still there. And I might even say some bits in Spanish at some stage, so we'll see how much is your knowledge of Spanish. Hello.
Hi. I've just lost my husband, but that's okay.
Anybody's got her husband? So today I'll tell you a little bit about the work I've done on pollination of New Zealand orchids, also about taxonomy, naming different species of orchids that we have in the country, and also the conservation work that one of my students at Victoria University has been doing for the last 2 years as part of his master's. So, as many of you may know by now, because you're interested in orchids, that this is actually one of the largest flowering plants in the world. There are about 25,000 species, and they have a worldwide distribution. Also, if you grow hybrids usually, or varieties of orchids, you will know that the number of varieties of orchids and hybrids, orchid hybrids, is actually way larger than the number of species that we have in the country, or sorry, in the world. Most of the orchids are epiphytic. About 70% of the orchids in the world grow on trees. And in those places where the temperature is rather miserable weathers and rather temperate, usually orchids are growing on the ground, so they are terrestrial. So that's what happens in New Zealand and also in South and South America. Something that I'm quite interested about orchids is that they have a highly specialized pollination system, and they have this very unique interactions with insects and less so with birds. Their life cycle is very complex. They depend on a mycorrhiza, on a fungal infection for seeds to germinate. So they make them very easy target, for example, or they can get easily threatened by activities that people, or habitat transformation, for example, or logging and other activities that affect them. As you can see from the photographs, they're quite diverse. You've got this tiny little New Zealand orchid over there, that's Drymoanthus. You've got these other ones that look like people hanging from their heads. And you have the flying duck orchid from Australia. So orchids, though, for scientists or for biologists are quite well known because of their connection with Darwin. And when Darwin was writing his book about the fertilization of orchids, he came up across this orchid, the comet orchid. This is a species from Madagascar, and when Darwin observed or studied the dry specimen, he saw this very long spur in there, and he thought that, well, this thing must be pollinated by a moth or an insect with a very long proboscis that allows the insect to collect the nectar that's stored at the very end of the spur. And it was only after 20 years of Darwin's death that this moth actually was discovered. Initially, it was given the name of Predicta because Darwin had predicted its existence. Orchids are quite important for horticulture, and also they have economical value. As you may know, vanilla is an orchid, and that's a quite important food flavoring, for example. Salep is also produced from orchid roots. This is a beverage that's used in many Mediterranean countries, and I understand that in the UK, when coffee was not as common as it is now, people were consuming salep. They also have importance in traditional medicine, especially in Asia, and one of the examples is Gastrodia elata, and you can see the tubers in there, and also Dendrobium nobilis, which leaves are consumed as tea. You can see they dry it and roll, and then you put them in your tea and have orchid tea. If we move into New Zealand orchids, we have about 110 or 100 species. It depends on who you talk to these days. And but what's very important about our orchids is that about 70% of them are endemic. That means they're only found in New Zealand and nowhere else in the world. We have only 9 epiphytic species, and plants, orchids in general, are found throughout New Zealand, even in some of the subantarctic islands, which perhaps is not the best place to grow if you are an orchid. We have 35 species of conservation concern. Again, habitat destruction is one of the main threats. Also weed invasion. And something pretty bad for orchids, but pretty good for me, they're quite understudied. So practically any work I do on orchids, it's a good contribution to advance our knowledge on this native group. So one of the aspects, when I first came to New Zealand, I was quite interested about pollination of orchids. And I was quite fascinated with these descriptions, for example, of Darwin's work. And I was hoping to find something very similar in New Zealand epiphytic orchids. Unfortunately for me, things are a little bit different here. And this is the work I've done for my master's. And I studied 4 species of epiphytic orchids. And all 4 epiphytic orchids are actually unspecialized. That means that they don't have these highly tricky systems with insects. And they're actually, the flowers are usually wide open. The nectar is present in there in most of them, just completely accessible for the insects to consume. We found out that about 20 species of insects overall visit these orchids, and only 4 flies were considered as putative pollinators. So that means that despite there's a lot of visitation, only a few insects were the right size to remove the pollinia and pollinate the flowers. This unspecialized pollination system is something rather common in the New Zealand flora. And if you look up there, many of our native plants, they have very small flowers. The flowers are wide open usually. And especially in the alpine areas, flight pollination is very important. So that was a bit disappointing, but then I moved into terrestrial orchids, and then things got a little bit more exciting. I studied some of the sun orchids, and as many of you might know, these things are self-pollinating most of the time. So again, things were a little bit boring, but then I moved into the greenwood orchids, and then things got very exciting. These orchids have this trap system in the flowers. So when the insect visits the flower, sits on the labellum, on the lip in here, and that closes very fast. And the insect is trapped in this chamber in here. The only way out for the insect is to go through the stigma and the pollinia, anther wings and the pollinia, column wings, sorry, and the pollinia, showing in there with the blue arrows. If you're lucky, you can get to see the insect sometimes stuck inside the flower. In about 30 minutes, the trap resets and opens, and the insect is allowed to escape carrying the pollinia. Why are these insects going to these orchids? We don't know. There's no nectar in there, there's no food for them. So we're thinking that they're using a deceptive system, so they're fooling their pollinators. Until today, we don't know why they're actually going in there. But another group in New Zealand that has an interaction with fungus gnats or mosquitoes, or these little flies, it's the spider orchids. And this is a group of orchids that I've been studying for the last probably 5 or 4 years now. So the very first record of pollination by fungus gnats and the genus Mycetophila dates from 1927 from a person down in the South Island. He was a high school teacher, and he did the very first observations of these insects on spider orchids. In the 1960s, it was put forward the idea that these orchids are mimicking mushrooms, both visually and in their odor. And in the 90s, they categorically said, well, these things are pollinated by brood site deception. That means that female insects are fooled by the flower and they lay their eggs on the flowers, and by doing that, they pollinate the orchid. Between 1979 and 1980, George Fuller up in Taranaki and in the Pukekohe Fernery did amazing observations on the pollination of the orchids. 2 species of Corybas that were growing in the fernery. And he identified several female fungus gnats visiting these orchids. Since then, no one has looked at the pollination of these orchids anymore, so I thought, like, that's a good project. And I started to study it. These previous hypotheses are completely opposite to what New Zealand Post is actually saying. They say that these spider orchids produce nectar, and that's what attracts the fungus gnats. These orchids do not produce nectar. There's no nectar at all in spider orchids. It's very similar to what we see in greenwood orchids. This is how the flowers look inside. You can see they're very small. It's about 5 millimeters. Same over there, 5 millimeters. You've got sepals and petals, a dorsal sepal, and the labellum in here, or the lip. Particularity of these plants is that they have this little opening in there, a little structure called the auricle, or perhaps exit hole or something, but all of them have these little holes in there. What's the function of that? We don't know yet. Something else, right in here, there's a little dip, a pouch structure, and that's where people has usually seen eggs and larvae alive and inside the orchid. This is how the orchid looks internally. You can see the column in here, stigma, and the pollen. And you can see that there's a sticky pad in there in the pollinia, and that's what helps the pollen to be attached to the thorax of the insect. Like I said again, there's no nectar in these orchids, and they don't have smell, at least to my human nose. This is the diversity of species that we have in New Zealand. The flowers are usually quite boring. They're green, or they're purple, or dark purple. They have just one flower, one leaf, and they're usually found in damp areas, leaf litter, and it's about 2 centimeters across, and that's again 5 millimeters, so that shows you how small these orchids are and how difficult to work with them actually can be. One of the species that's one of the most common spider orchids in New Zealand is Corybas trilobus. It's quite widespread, and it's also quite variable morphologically. Their shapes is very different. And this is the species that I've been studying for the last 3 or 4 years. The specimen over here was collected by William Colenso in 1847, and that was the very first record of this orchid. Those specimens were collected in the Wairarapa and Cape Palliser, and at the moment the specimen is at Kew Botanical Gardens, but I've managed to ask Kew to send it to me to study it, so that's been pretty useful. Although you can see that after you press the plants, the flowers are completely distorted and you cannot see anything. It's just like a blob in there. So that's my plant, and one of the different forms of Trilobum actually. What you're seeing here is the whole variation of the species. And Nematoceras trilobus has been considered a species aggregate. That means that all these other things are lumped or aggregated under this name, Trilobus. And many of these things, these are only 8 of the different forms. There are up to 25 tag names recognized up there. They've been given very funny names. We got Nematoceras trilobulus. This is round leaf because the leaf is slightly rounder. Tri-dot because it grows in Dot's farm. Tri-white because it's slightly whiter but rather yellow to me. Darky because it's darker. We've got Eastern Hills because it was collected in the Eastern Hills of Nelson. And Trotters from Trotter's farm as well. So there are several of these entities out there that we don't know if they're actually real species or just represent variation within the species. And the Department of Conservation actually has been quite interested in these entities because some of them are very uncommon in the country. They're just found in a few spots of New Zealand, and therefore they're looking to see if they actually need to invest money into their conservation or not. So when I started this project, there were some of them quite interested in the outcome. These are some of the places where I've worked and I've collected. This is in Kahurangi National Park, in the tablelands in here, and in the South Island. And you've got Nematoceras tri-white up there and round leaf down here. You may notice that sometimes talking about Corybus, and then I moved into Nematoceras, and that's a problem of taxonomy. When I started the project, everything was Nematoceras, and now that I finished the project, everything is Corybus. So I still do. And that was about just a few years in between. So when I first arrived into New Zealand, everything was Corybus. I left the country, came back, everything was not Nematoceras, and keep changing. I'm not going to change it. So. This is the habitat of Nematoceras darky. This is in Nelson Lakes National Park. Luckily for me, the plant grows down here underneath beech forest. Usually we find forget-me-nots and buttercups up there. This is the only place in the country where Nematoceras darky grows. It's nowhere else. So it's a very unique form and with a very restricted distribution again. And this is Nematoceras Eastern Hills. And that's my backpack, and that's the habitat where it grows. Again, beech forest, and usually among leaf litter. Some rather more inhospitable habitats. This is Nematoceras tri-arth, because it's from arthrospaz. And I'm actually went up here, so this is the Ochira. And you go up here, and that's where the populations are. It's very close to the snow, it's very high, it's very damp. It was a very miserable day. I was actually looking for forget-me-nots, and that's Mount Cook lily buttercup in there. I couldn't find the forget-me-nots, so completely disappointed. I just looked to the ground, and I saw the orchids flowering in there. So that was a good saver for the day. And it was a form that I didn't have in my previous collections as well, so that was good. So as part of this project, I also had funding for a master's student, and this is Chow Ping On, who's been working mainly in the pollination aspect of the project. So I was doing work on taxonomy and systematics, and she was looking into the pollination. And basically what we wanted to know was what species of fungus gnats are visiting these orchids. And you can see her, she's doing some notes in the field, and we also use some paper clips. You can see the very small leaves, paper clips with numbers, because we were tracking the development of the flowers to find out how long does the flower take to open from bud, how long does it take until after we pollinate the orchids for the fruit to develop, and then how long does it take until the capsule is mature and releases the seeds. And we also wanted to find out if they really needed the insects or not for reproduction. And for that, we devised this system that it's just a plastic container with a tool over it, and we just place it right on top of the orchids. And in that way, we excluded any insect that was trying to visit the orchid. And we could test if they actually need the fungus gnats or not. And our results were show that they actually do need the insects. If there's no pollinator, they don't produce seeds. So they depend completely on these insects. We also studied the behavior of these insects when visiting the orchids. And for that, we use this night vision cameras, and it's just a typical surveillance camera that you might see in some buildings, and it's just connected by a very long wire to this black box. Inside the black box we had a battery about this big and quite heavy, and a memory card, and every 12 hours we would come to the site and replace the battery and replace the memory card. And then Xiaoping would spend lots of hours looking at these videos, which is something not very exciting because sometimes nothing happens for hours. And we also looked at the diversity of insects or fungus gnats in these habitats, and we used Malay traps. And these traps are just very simple. It's a black tool down there, and the top they have a white tool. And the idea is that the insect is just flying, minding its own business, and then crashes into the tool, and then see brightness up there, you think that must be the exit. So they go all the way up, and they start to crawl towards up this part of the trap. It just looks like a tent. And there we have a flask with ethanol. That's the last thing that they will see when they reach. So it's not very, don't always go to the light. You don't know when there's a train coming. Well, in this case, it's just ethanol. So we managed to get quite a lot of samples from there that Xiaoping had to identify to the species level of fungus gnats, or find out what species they are, so then we could see what is around these orchids. To our, if you've never seen a fungus gnat, this is how they look, and sometimes that comes out as fungus gnats, and it should be net or something like that, but just the accent. You can see they're quite small. That's 5 millimeters there, 5 millimeters. And what we found out is that both male and female fungus gnats were actually visiting these orchids, which sort of like extends a little bit more to what was previously reported in the literature. The yellow bits that you see in there, that's pollinia, that's the pollen of the orchid. And if you wonder how you tell apart a male from a female fungus gnat, well, you just have to have a very good microscope and look at the bum of the insect. The female fungus gnats usually will have a pointy bum towards the end in here, and that's where what they use to lay their eggs. And the males will have less pointy abdomen, and at the end they will have clasps which help them to secure the connection with the female when they're mating. So in this table we have all the different forms of orchids, the different species of fungus gnats, and the gender. And as you can see, we have both male and female. To identify the species of fungus gnat, in this case we have only AF, and that means that the entomologist was not very confident with the identification. And that's because sometimes many of these insects are, when you're handling, you may damage their abdomen. And if you have, and the abdomen, especially of the males, it's quite important to identify the species. And actually, to identify the species, you need to look at the male genitalia. If you don't have a male, if you capture lots of fungus gnats, only those that are male are the ones that you can identify to species level, because the description of these insects was based on the shape of the genitalia of the male. So females, we cannot identify them to species level most of the time. Something else that we discovered from this work was that these insects actually are using the flowers as a mating ground. And you can see that's a male in there. And male fungus gnats usually sit on the flower and start this wing vibration dance. They move the abdomen up and down, and you can see that's a female over there who's very interested in the male. They mate, and the affair is very short-lived. It's just a few seconds, and then she looks for a place where to lay the eggs in the flower. Meanwhile, the male is calling for another female, so it's just like that. And you see the female keep looking for a place, and we think that when the female is trying to find a place to lay their eggs is when pollination happens. Now this is a second clip, and we can see now a bigger male, and this the tiny little male in there. And you can see again, there's wind vibration, the abdomen's moved up and down, and soon you'll see that his moves are quite successful and must be a very cool dancer, because you'll see a female arriving pretty soon. We believe that probably these males are, when they're moving the abdomen up and down, are releasing a pheromone, and that probably attracts the female fungus net. Now, they usually do this on mushrooms. So we think that probably these orchids also are scented as mushroom, or they mimic the smell of a mushroom. You can see the mating over there, and the other little male over there is quite angry, and there's a bit of a, you see there's a bit of a fight in there, and kicks the female out, and later on he will kick out the male as well out of the flower. So it's been, this is about, I guess it's about 1 minute 53 of footage that I'm showing you. And it was hours and hours of filming. So it's something very, it's not like you go and look at them and everything is happening in front of you. It happens in, it's not that common.
So it's the female that dies, is it?
What's happening there? So I'll just go back to this. I can't find the screen. Do you know how it is? Sometimes. Okay. Oh, sorry about that. So, this is what people has reported before from these flowers, but people from the orchid group, for example, that have in the journal, you'll go through their journals and you will see that they found eggs and larvae. No one knew what these eggs and larvae were, belonged to what species, and well, it was thought that it was to fungus gnats. And usually you find these eggs among these hairs on the labellum in there, or in the egg pouch over there. If you're lucky, you might see the eggs actually hatching and the larvae just moving around. There's no food for this larvae in the flowers, so they just die, or probably they might leave the flower through the exit holes. We don't know. As you can see, there's no way to tell to what species these things belong to, because there's no features in their shape, for example, or in their colors that help you to identify them. They are white and they just have a black head. So we've been using DNA analysis to identify these little maggots and larvae. And in this figure, what I'm trying to represent is the similarity of genetic similarity of the different samples of eggs and larvae that we collected from different orchids. So we have 6 different forms of spider orchids in here, and Xiaoping got some of the eggs and larvae, crushed them, got DNA out of them, and then we put all our DNA sequences together, and we looked at how similar or different these different samples were. And that's when things got a little bit exciting for me, because we had, for example, eggs and larvae from this species, Nematoceras hypodium, from Te Urewera National Park and from Nelson Lakes. And you can see the eggs, that genetically those 2 egg samples from 2 parts of the country are grouping together, and that means that they're practically identical in their DNA. If we look at this other form up here, that's from the form Pygmy, eggs and larvae from Gordon's Park Reserve in Wanganui, and also from the Sutherlands in Rangitikei. And again, those 2 are grouping together. So that means in those 2 different parts of the country, or well, in the Manawatu area, this same orchid is being visited by the same female fungus net. You can see we have M. filicornis in there, and a number that says 100 in there. That means that in many of our analysis, there's 100% support for those sequences to be related to this species. And so that we can assign actually a name to those eggs and larvae. We can say those are the eggs and larvae of Mycetophila filicornis, and we have 100% support for that. What you can see in here that sometimes I just don't have a name, I just have a bit of a code in there, and that means that for those sequences we don't have a species name, because first we need to capture a male fungus gnat, which the entomologist will give me a name. Then we get DNA out of that male fungus gnat, and then we compare the DNA of that male fungus gnat with all our eggs and larvae, and then we can give a name to the egg or larvae. But what was interesting is that we were having this sort of like a specialization towards one species of fungus gnat. And then we looked a little bit more, and we wanted to find out why these insects are actually visiting these orchids. Do they actually smell like mushrooms? And do all forms have a different aroma? So what we did, we started collaboration with Andrea Sella and Rob Casers, which are chemists, and this is people working at Victoria University. Rob is quite interested in aroma, especially the scent or aroma that you get in wines. So he's been doing a lot of wine analysis of Chardonnays and Sauvignon Blanc. So using his skills and Andy's interest in spider orchids, they started to work on this project. Every single point in here represents a sample from the form Remutaka, for example, 4 samples from that form, 4 samples from that form, etc. If the points are very close together, it means that they're very similar in their smell. If they're very apart, it means that they're different. So what we had from this preliminary analysis is that this there are 6 different forms of spider orchids have different aroma. And we're thinking that that's the mechanism that they're using to attract different species of fungus gnat. Now, how do you do a scent analysis? Well, it's quite easy after you find out how does it go. This is a flask in there. We have our orchid in there, just at the top. It's got a rubber septum or a rubber tip where you put a needle that goes through it. And the needle captures all the aroma or all the molecules that are on the air, accumulated in the air inside of the flask. And then they go through this machine. The machine separates all the different molecules inside of that create the aroma, and it runs it from 4 minutes to up to 30 minutes. And you can see there are like peaks every certain minutes in there, and that means that the machine detected a compound. Then, for example, if that minute 20 you have a compound in there, the machine will look into a database and we'll see what kind of compounds are normally detected at the minute 20. So then at the end we get a list of different chemicals or molecules inside of the aroma of these orchids. And we found some mushroom-like content, aromas, but like I said, it's a very preliminary assessment. I need some more years of work on this. Now I'll just move to Gastrodia, to potato orchids, and this is a project that I've been working on for the last 2 years in collaboration with the Department of Conservation, Jeremy Rolfe in particular, who's been quite interested in finding out the number of species of potato orchids that we have in the country. As you can see, they're very dull, non-colorful orchids at all. They're practically brown. No chlorophyll, so they don't have any they don't photosynthesize. And there's no leaves, just that tiny little bract in there. And one unique feature of these orchids is that the lip is at the top of the flower. And that means that the flower is not resupinated as many other orchids. Usually orchids will have a torsion of the ovary, and that will move the labellum to the lower part of the flower. And that's sort of like the very unique feature of orchids. We have about 50 species of Gastrodia in the world. Like I said, no leaves. They're usually found in moist or sheltered forest areas. And initially I thought they were found only in Australia and New Zealand, but found out that they found also in Asia and South Africa and and the Southwest Pacific. They're not very colorful. This is the diversity of Gastrolia in the world, or well, in some parts of the world. You can see that that's the same pattern that we have in New Zealand, rather brown. This is the most colorful Gastrolia that you will see out there, and this is Gastrolia javanica from Java, Albidioides from Taiwan, and Gastrolia rwandensis from Africa. Unlike our New Zealand Gastrolias, the labellum or the lip here is at the base of the flower. This is Gastrolia similis, and it's a species that grows in Reunion Island, just a tiny wee island that belongs to the French next to Madagascar. And I was lucky enough to visit Reunion Island a couple of years ago, attended to a conference over there, and I met Florent Marteau over there, who's a scientist working on the pollination and the mycorrhiza of this orchid. And after that meeting, now we've started a collaboration project in which we want to find out how Gastrolia from Reunion Island, for example, are genetically related to New Zealand Gastrolia. But he has also been working on the pollination of these orchids, and that little thing that you see up there, that's the head of a fruit fly coming out of the flower. This is pollination in Gastrolia, and as you can see, labellum is down here. Again, these orchids don't have nectar. There's no food for the insect in there. And from the work that Florent is doing, he found out that they're actually fooling their pollinators as well by producing the aroma of fermenting fruit. So the same kind of analysis, chemical analysis of the aroma that I'm doing, look at that. So they fool also the female fruit flies. In this case, there's no eggs inside. These flies are not depositing egg or laying egg inside the flowers. That's 4 minutes later. I'm not sure if it's the same fly or a different fly, but you will see that this time things go slightly different. And it's a different trap mechanism to what we have in the greenwood orchids. That's very fast. So you can see it just go gradually moving up. And now there's practically no exit for, no escape for the little fruit fly. And look at that. See, and then removes the pollinia. So the only way that now the poor thing is going to get rid of that is by visiting another, not spider orchid, Gastrolia orchid, and pollinated. You can see it's trying very desperately to get rid of the pollinia. And he's got, that's 24 minutes later, and he's still trying hard. And yeah, so the video will finish, and the very end still's got the pollinia attached to the back. So, like I mentioned before, Gastrolia is quite important for traditional medicine, and as you can see from the slide, they're practically used for anything or everything. Stomach disease, improve memory, prevents stroke, headache, diabetes, etc. How much of this is supported by scientific tests? I don't know, but that's what's out there. What people do is usually they go out in the bush, collect the tubers. The big ones are sold or dried and used as powder or tea. And the little ones are grown in little pots like you see there. This is a very proud farmer over there with his plantation of Gastrolia. Gastrolia in Asian medicine is actually, or Asian countries, is widely used. And nowadays you can find Gastrolia ice cream. You see the tubers in there. If you have problem with wind, you can get those peels in there. You can use it on chicken. You can drink Gastrolia tea, or if you're worried about conservation, you can use these eco-friendly Gastrolia tubers. You can buy them for about $60 US per kilo. This is Gastrolia canningamia. It's one of the most common gastrolias that we have in the country. And I should say as well that Maori has used gastrolia as well. They used to dig them out and eat them or roast them or use them in hangi. And I've heard of people that still eat some gastrolia tubers down in Invercargill. There's a professor, the teacher, that takes his students to try gastrolia tubers. Gastrolia canningamia is usually found in beech forest. Gastrolia minor in Kanuka or Manuka scrap, and this is how the tuber looks like. So it's a it's the size of my little finger. And that's a plant that's starting to develop. They practically look like asparagus when they start developing. And sometimes you find them in the city as well. This is Gastrolia minor growing in Lower Hutt in one of the traffic islands. And this is Gastrolia canningamia growing here in the Esplanade Gardens. If you don't know where it is, that's the population in there. So despite being a very picky plant or species, they're practically found nowadays in several disturbed areas. Another species that's normally found in disturbed habitats is Gastrolia sesamoides. And I just put the sesame plant illustration in there because Gastrolia sesamoides was named after this plant because they're very similar according to the taxonomist that wrote the description. I can't see much of a similarity between those two, probably because of perhaps the capsules are slightly similar, I don't know. But the name Gastrolia means actually, or relates to stomach, or it comes from like gastronomy, Gastrolia. It's because the flowers are have the shape of a stomach. And this is how the tubers of Gastrolia sesamoides look like, and they're definitely bigger than the Gastrolia minor. So there are two other entities within Gastrolia that DOC doesn't really know, or taxonomists out there don't know if they're different species. One of them is Gastrolia long column. The column inside the flower is long, and it's found in several parts of the North and South Island, but normally normally found in disturbed parts of the disturbed vegetation or disturbed habitats. And this is Gastrolia long column black. Again, a long column, and in this case the tip of the labellum is black. And unlike the other orchids or the other Gastrolia in the country, there are only 3 populations of this orchid in the country. One was recorded in Wanganui area. My points are a little bit out of place. Another one in the Wairarapa, and just recently this year we found one in Nelson. The site in Wanganui, it's gone. We haven't been able, it was reported about 10 or 20 years ago, and we haven't been able to find any plants in there. So that means there are only 2 parts of the country where we can find these orchids. So is this actually a different species, or just a variation within Gastrolia canningamia, for example, which is the most common potato orchid? We got, that's Jeremy Rolfe and Jasmine Gibbons. We got funding to get a summer research fellowship and a student from Victoria University to work at the museum and also at Victoria University on this orchid. And what Jasmine was doing, she spent the whole summer measuring plants and also did some work in the lab. She measured about 250 specimens, and after she finished this project, she said she was going into cancer research and no more taxonomy. I don't know if that actually put her off from plant science or what. But after she did all this work, unfortunately morphology didn't help much, and we couldn't, besides the differences in the inside of the flower, we didn't find any major differences. So we looked at the DNA analysis, and again, what I'm showing here is different samples, for example, of Gastrolia sesamoides, which is down here. Things that are very similar genetically are grouping together. Things that are genetically distinct are far apart in the graph. So down here I've got all my samples of Gastrolia sesamoides forming a single group, that's pretty good, it's a good species. We looked over here, we have Gastrolia long column by itself. It was like, oh yeah, we've got another different distinct group. Here we have Gastrolia minor, the little potato orchid by itself. But when we went to Gastrolia caningamiae and Gastrolia long column black, we couldn't find any difference in their DNA, in that part of the DNA that we looked. So then we went and looked into another part of the DNA of these potato orchids, and this is what I'm showing here. And again, Gastrolia caningamiae and Gastrolia long column black are identical. So genetically, we don't have enough support to actually say these are two different species. I wasn't happy with this result, so I went and I used another technique that looks a little bit into more detail, and it's a kind of fingerprinting technique. And it's a technique that you will use, for example, to identify who's your father or who's the person that committed the murder. And this technique, what it does is just picks differences between organisms that are very closely related. And I'm showing different species of potato orchids in here by different colors and shapes. And we've got Gastrolia minor, for example, over there, Gastrolia long column in here, and I've got long column black and caningamiae there, just very close together. I'm still deciding what we're going to do about that because they're very, very, very close together. They're not, probably I'll have to talk to Tricia or to Richard later to some advice, but they're so similar that I don't know if we can actually use genetics to say they're different species. And this is, well, that's what I said in the next couple of months, I'll just have to think about what do we do. Is this actually a different species, or is it just a variety? If it's a variety, probably we have to describe it as a subspecies. And, or it's just a single species that have these two types of flowers. And this is how the plants actually differ in their internal structures. You see the column is right in there, very short, it's a sulfur, and the other entity, it's an outcrosser, very long column. And you can see some differences as well in floral shape. So it's probably a very recent speciation event. I don't know yet. Their pollination is not as exciting as the ones from Reunion Island. Many of the species are sulfurs, and you usually see spikes full of capsules, all very fat, full of seeds. The species that are outcrossers are visited by this solitary bee. It's a native bee, Lasioglossum sordidum. There's no nectar in these orchids, but they produce this powdery substance in the labellum. And we believe that that's what the bees are collecting from the flower. This powdery substance or grains are termed or are called pseudopollen or fake pollen. And when you look under the dissecting scope, you can see that they have tiny brown, these tiny little dots inside the cells, and that means that they have starch in the cells. So that's probably what these bees are collecting from the orchids. And now towards the end of my talk, I'll just tell you what I'm doing, or my student has been doing, on the conservation aspect of these orchids. Like said before, these orchids have this fungal interaction, and it's, they have a mycorrhiza or an endomycorrhiza. And mycorrhiza is a fungal plant root association. All orchids have this association, and they're really needed for seeds to germinate. If you don't have the fungal infection, the seed will not germinate unless you're using asymbiotic methods, which are using lots of chemicals and other nutrients. The orchid gets access to organic and mineral nutrients. What the fungi is getting out of the orchid is unknown. And there's quite a lot of studies out there that have tried to answer this, and it seems that actually the fungi is being fooled by the orchid. This is how the hyphae sort of like accumulates or grows inside of the root. This is a root cell, and you see these little pelotons, that's the hyphae. What's interesting about this interaction is that it's usually species specific, and that means one species of fungi to one species of orchid. orchid research in fundamental. Yeah. So it's, orchid endomycorrhiza actually has not been widely studied in New Zealand. The very first studies are coming from Dame Ella Campbell at Massey University, and she studied the endomycorrhiza of 3 species of Gastrodias in the 60s. And then we have Roger Watkins in 2012, who's done some work in spider orchids. But despite these 4 studies that we have in the country, no one has looked into seed germination symbiotically, using the fungi that naturally find inside the orchid roots. And because of that, we don't have restoration efforts that are looking into reintroduce orchids into habitats that have been restored, for example, or in urban protected areas. And we don't have ex situ conservation. We don't have collections, for example, of native orchids in many of our botanical gardens. This is what happens at Kings Park and Botanical Gardens in Perth, and what you see in there, it's many, many seedlings that they have grown symbiotically. That means with its fungi. So to develop this kind of projects, I managed to get funding from different organizations, Australian Orchid Foundation, San Diego County Orchid Society from the States, also the Wellington Botanical Society and the Otaria Wilton's Bush in Wellington. And we put all this money together and collaborating with, put some collaboration in place with Perth, with Kings Park, and my student Jonathan Frericks went over there and learned all these techniques, and then came back to Victoria University and started to work with this, with orchids under the supervision of 2 scientists over there. He had 3 main goals of his project. First was to isolate and identify the fungal partner of 6 terrestrial orchids. He wanted to implement these methods here in New Zealand orchids, and also we wanted to find out or evaluate seed germination success at different distances from an adult plant. If you got your seeds, will you plant them right next to the adult plant, 10 minutes, 10 centimeters away, 20, a meter, etc. So he was trying all these different trials. Today I'll tell you about the most exciting success that he had in his project, and this is Aspiranthes novaezelandiae. It's a nationally vulnerable species, and as you can see following the arrows, we were quite lucky that everything worked fine, and we have now about 6 plants of this orchid. The process that you have to follow to get to this plant over here is first, unfortunately, you have to sacrifice a couple of your individuals of your threatened plant, cut bits of the root off, and then tease out the pelotons. These are the little things that I showed you before that were inside the cell. The peloton then is transferred to this plate of agar, and you leave it growing in there for a couple of days or a week, and you can see it's starting to grow in there. You use part of that growth for DNA barcoding, so you can identify the species or the strain of fungi that it's growing on that plate. And then you cut a little bit of that gel and inoculate your seeds, and then you just wait. 110 days, we got the first leaf, and you can see that it's already green, so that means producing its own food. And 230 days later, it's 4 or 5 flowers, leaves, sorry. The plants, we still have them, but they haven't flowered yet. It looks very simple and easy to achieve, but it's not, because he found out that it's not necessarily, you don't always find one species of fungi or fungus inside the roots. And in some of the orchids, actually, he managed to find up to 16 different entities of fungi growing in there, or entities of fungus. And to find out which of all of those are the ones that promote germination, you need to do the germination experiment with all of them. So, and he had 6 species as well, so he had 6 times 12, and then etc. From all the different combinations he used of species and fungi, this is the only one that worked. He was very disappointed, but I was quite happy because it was the only threatening species that we had in our experiment, or his experiment.